Hello, everyone. Welcome to our webinar, Innovations in Drug Discovery, Advancements in Miniaturized Protein Engineering and Mass Spectrometry. Thank you for being here today.
My name is Neela Lay. I'm the marketing manager here at DISPENDIX, and I'm very proud to be moderating this webinar today.
On today's agenda, we will hear from an exciting panel of experts in protein engineering and mass spectrometry. Please feel free to use the questions feature throughout our program. We will have 15 minutes at the end for a Q&A. Let's get started. The first panelist is Leann Teadt from Codexis.
Leann has emerged as a key figure in protein engineering. With 5 years of dedicated service at Codexis, she serves as a scientist in the protein engineering group. Leann specializes in optimizing high throughput screens and analytics. Her pivotal role extends to technology development, where she actively contributes to innovation and process improvement. Leann's commitment to advancing protein engineering has positioned her as a leading force in the biotechnology and pharmaceutical sectors, leaving an enduring impact on scientific progress. She received her PhD in chemistry from Emory University under the guidance of Dr. Stefan Lutz.
Thank you for the introduction, Neela. I'm going to talk about a couple different projects at Codexis using enzyme engineering, both in a technology development standpoint and looking at some increasingly complex protein engineering problems that we're hoping to solve. So to start off, Codexis works by essentially reimagining enzymes. We take enzymes from nature, we apply our CodeEvolver platform to create commercially relevant biocatalysts. These biocatalysts can then be used in a variety of industries, such as pharma manufacturing, and more recently, RNA manufacturing. The way our CodeEvolver technology platform works is we identify a protein with some sort of starting activity. We then iterate, design, build, test, learn cycles until the protein is ready to bring to market for whatever purpose it's needed via scale up and production. So for my talk today, I'm going to start by talking a little bit about some technology development work that we've had, and then I'll briefly talk about our ecosynthesis platform. So here at Codexis, we are constantly working to improve our technology, and one of the ways we've done so recently is through miniaturization of some of our workflows. So, why would we want to focus on miniaturization? There's a variety of reasons. One is just time. Miniaturization can allow for a faster workflow. It can also allow us to increase our high throughput screening capacity. And this can allow us to do more multi-parameter screening a little easier. And then with higher screening capacity, we can actually employ more data-intensive approaches. As we can screen larger data sets, those can be then used to train predictive algorithms or to do more deep screening, which can feed back into machine learning and further improve our technology. And then on a more practical level, miniaturization means less reagents and solvents, which is less waste, lower costs. It's also a more sustainable option in many ways. There's fewer consumables, especially less plastic. And then, everything's miniaturized, we can take up less space and have a reduction in our lab footprint. So there's a lot of different ways to miniaturize. And the way we chose to go forward after looking at lots of options was through surface patterned microarrays. And these are glass surfaces with hydrophobic and hydrophilic patterning on them, which kind of allows us to take advantage of the so-called lotus leaf effect. So you can see here on the screen, these arrays are about the size of a standard microscope slide. And on this array shown, there's more than 1,000 droplets, and they are able to form aqueous solutions in very discrete, stable droplets, ranging from about a 10 nanoliter to 150 nanoliter volume. So that's the reaction volume at that point. And the nice thing about having such dense microarrays is in the footprint of a traditional microtiter plate, we can fit more variants than we would use in a round of evolution, just essentially in one small space. One of the things that we immediately ran into in looking into these microarrays is how to get liquid on and off of them in a reliable manner. We kind of made a list of everything that was important to us, what we needed, and this is actually how we came upon the I.DOT. It hit our major points the best. One of the key criteria for us is that we needed to be able to add a broad spectrum of liquids to these arrays. Our workflows use anything from organic solvents like acetonitrile to aqueous buffers, solutions with 50% glycerol, cell suspension. So we really needed something that could work for all of that. We also needed something really that could handle parallel liquid dispensing. Like I mentioned, there's over 1,000 droplets on that one array. So if we're dispensing them individually, it's going to take a lot of time. So we really liked that we could do, 8 dispenses at once with the I.DOT. It actually enabled some of our workflows. We also needed it to be adjustable to different microarray densities. We didn't want something that would only work for one specific density of microarray. We wanted to be able to switch our densities and also use it in a traditional microtiter plate format. We wanted a more versatile instrument. Once we kind of got a lot of these logistics worked out, we wanted to be able to test whether or not we could actually run an evolution on these microarrays. So we wanted to put this theory into practice, and we did this through the evolution of a DNase. DNase is a genotoxic protein. It's not easily expressed in vivo in traditional E. coli platform, which is what we would typically use. So we really liked that we were able to express this in our cell-free protein synthesis platform on the microarrays. That kind of let us use a couple new technologies all at once. And then also DNase is likely to be used in extremely small volumes, so it makes sense to evolve it in that context as well. When we looked at how to evolve this and monitor the reactions, we looked at a fluorescence assay that would give us a nice output for our DNase activity. So the way this assay works is there's an oligonucleotide with a fluorophore on one end and a quencher on the other. When the oligo is intact, the quencher is close enough that no fluorescence is emitted. As DNase is active and cleaves that oligonucleotide, the fluorophore is released and emits fluorescence. This whole reaction can then be stopped with EDTA, which inactivates the DNase. We were evolving for a couple KPIs, specifically enzyme stability and specific activity in the presence of an inhibitor. Since this is a new platform, the microarrays, we also carried out all of this work simultaneously in traditional microtiter plates just to benchmark our work. And over the course of 3 rounds of evolution, you can see on the graph here, our lead variant ultimately showed about a 23-fold improvement in activity under the relevant conditions in the presence of an inhibitor. We also saw a 7-fold improvement in thermal stability. And as we went through the rounds of evolution, we did see similar levels of improvement within each round between the microarray platform and the microtiter plate platform. There is significantly worse signal to noise in the microarrays. There's a couple of reasons for this. One is those droplets have no walls between them. And this is a fluorescence assay, so we do get some crosstalk between droplets, which added to our noise. Additionally, the microarray platform, we found that at the very small volumes, the reaction was extremely fast. So we actually had to limit it to a 1-minute assay. This is where that parallel dispensing that I mentioned actually enabled this assay, because we needed to be able to dispense everything on the slide quickly and then quench it within a minute. And we were able to do so, which is exciting for us. The microtiter plate, just for reference, was about a 10-minute assay. So a little bit different kinetics within the volumes of the reactions. So then when we actually finished the evolution, we looked at how the diversity correlated between these 2 platforms. And what we saw was that overall there was pretty good correlation between them, especially in the beneficial diversity that was picked up upon. The most beneficial mutations all matched really well. There were some areas where the 2 platforms didn't agree. They had slightly opposing performance. This is mostly due to that high signal to noise in the microarray, as well as the fact that they were slightly different conditions that they were being evolved under. So it's not too surprising that they're not a perfect match. But we were really excited about how well the diversity did correlate and that we could evolve an enzyme in this microarray format. It was a nice way to look through this technology and see it work. So I'm now going to actually shift gears, and talk about our EcoSynthesis platform that we're developing at Codexis. The EcoSynthesis platform is positioning us to deliver in the RNAi market. I'm sure you guys all are aware of RNA therapeutics, especially after the pandemic. They feel almost ubiquitous. It's been a great emerging technology. With the RNAi demand that is coming down the line for these therapeutics, chemical synthesis alone really is not going to be able to meet this projected future wave. Customers have been asking us for a scalable, more sustainable enzymatic solution to complement that chemical synthesis. The reason I mentioned sustainability is phosphoramidite chemistry uses a ton of acetonitrile. You can see on the slide, a kilogram of RNA requires about 1,000 kilos of acetonitrile to make it. That's not ideal. We'd like to kind of shift away from some of that solvent use. Codexis is using our 20-year history of enzyme engineering and expertise in pharma manufacturing to deliver this enzymatic solution via the EcoSynthesis platform. And EcoSynthesis is enzyme catalyzed oligonucleotide synthesis. And essentially what this means is it's iterative single nucleotide extensions of an oligonucleotide. So there's an initial substrate starter oligonucleotide and a 3' phosphate blocked ribonucleotide. They're joined to form an extension product, then deblocked to remove that 3' phosphate. That gives the N+1 product of the oligo, which then becomes the new substrate oligonucleotide to iteratively extend the sequence that's desired. So in addition to the enzymes that are needed for the extension and deblocking, we're in the process of supplying an enzyme cascade for the synthesis of the 3' phosphate blocked ribonucleotides, which also generally have 2' modifications at them. We're calling these NQPs. And then also the enzymatic synthesis of the starter oligonucleotide. There's a ton of enzymes involved in this process. I am going to kind of focus on just one of them for today, and that is the terminal deoxynucleotidyl transferase, or TDT. This is the enzyme that's catalyzing that extension reaction. This enzyme has a few KPIs. One is obviously just robustness, manufacturability. We need to be able to produce it, and people need to be able to use a relatively stable enzyme. Another target is high productivity. So phosphoramidite chemistry is great. Very efficient. So we also need to be extremely productive and have over 99% conversion at millimolar substrate concentrations. And then finally, this enzyme needs to have a great substrate tolerance and promiscuity, both accepting modified ribonucleotides, which I'll get into in a second, and have minimal oligonucleotide sequence bias. The enzyme really sees about the last 3 bases of the oligonucleotide, and it needs to accept any modifications there. Looking at some of the substrates that TDT could see, the nucleotide substrate scope that it needs to very efficiently incorporate could be any relevant base, various relevant 2' modifications, either a 2' fluoro or methoxy group are what we're currently investigating. And then it needs to accept a 3' phosphate blocking group as well. And then in some cases, an alpha phosphorothioate bond, either on the NQP or in the oligonucleotide that it's seeing. It needs to recognize all of that on the NQP side, and then additionally, those can all be incorporated in different orders in the oligonucleotide that it's seeing. So it needs to be fairly unbiased towards that oligonucleotide as well. So we're asking a lot from this enzyme, and our engineering team has done a fantastic job developing great screens for this. So screening efficiently becomes key for this enzyme. And there's a few different tricks that they've utilized. One is miniaturization of the screening assay down to 1 microliter or below. This enables a lot of these honestly fairly expensive reagents to go a lot farther in screening. They've also managed to multiplex conditions within a single screen so they can look at a bunch of different parameters all at once. And then finally, they've made it a robust assay over thousands and thousands of variants. And this is, again, where the I.DOT has come in super handy for our screening team. It's really kind of become a workhorse for them. It's been great. So looking at what this program has done, just to kind of let you know, this is one of the most complex evolution projects that's been undertaken by Codexis to date. It's a very complex screen and target. So here on the left of this table, you can see these would be the last 3 to 4 nucleotides of the oligonucleotides that the TDT enzyme is seeing. And then these are the various ribonucleotides that would be added. A 3P is the 3' phosphate blocking group. An M would be that 2' methoxy. An F is the 2' fluoro. All of the different RNA bases, as well as the asterisks, indicate that phosphorothioate linkage. A huge variety of compounds that this enzyme is seeing, and initially it really didn't work well on anything. Either it didn't work at all or it was very minimally active. These are percent conversions in high throughput. However, over iterative rounds of evolution, the project team was able to get it to increase its promiscuity and then increase its conversion to where, at the far side of this table, you can see all of these have conversion and most are above 50% or higher. The team is still working on this enzyme. This is an active evolution and working on improving the incorporation efficiency of each of these nucleotides to more than 99%. However, even now, the project team was able to use the TDT that they had evolved to announce the achievement of a gram-scale oligonucleotide synthesis with this ECO synthesis platform. We were really excited to be able to announce that back in December, and the team did a great job achieving it. And so with that, I would like to thank everybody who's been on either of these projects. This was very much a group effort in both of these cases. So with that, I'm gonna pass it back to you, Neela, and thank you.
Thank you so much, Leann, for that insightful presentation. I hope you're enjoying the webinar so far. Our next panelist is Jack Cahill. Jack is a distinguished R&D staff scientist at Oak Ridge National Laboratory, where his impactful contributions have spanned 9 years. Armed with a PhD in analytical chemistry from UC San Diego, Jack has cemented himself as a leader in the realm of mass spec techniques, specifically focusing on the cutting-edge development and application of the next generation for the chemical characterization of intricate systems.
All right, thank you very much for the introduction. I'll be talking today about a similar topic as Leann's of enzyme engineering. I'll be kind of giving more of an intro of how we developed the technology to enable us to do some of those things at Oak Ridge, and then some of the results that we've gotten. As Neela mentioned, I'm at Oak Ridge National Laboratory. I'm part of the bioanalytical mass spectrometry group here at the lab. We're part of the biosciences division. This picture is of myself and Vilmos Curtis, who I work very closely with on all of our mass spectrometry and instrument development works that we do. Our group is really pretty well established now in developing new technologies for mass spectrometry system development. And we mostly use these techniques for high throughput chemical analysis or just chemical analysis of difficult to measure systems. And at the National Lab, we have projects that range across the gamut of different fields. And so our work spans a range of polymer chemistry, materials chemistry, soil chemistry, biochemistry. We kind of spread these techniques over a lot of different areas. And a nice thing is that many of these technologies have eventually become commercialized in various forms. What I'm going to focus on today is our open port sampling interface mass spectrometry system. And so this is an approach that was developed by Gary Van Berkel, who was the group leader of our group several years ago. He developed this technique to enable online liquid extraction of any droplet or any sample coming in contact with this OPSI MS probe. And in essence, how it works is that it's a coaxial capillary system where we're flowing solvent in the outer channel. That inner channel is connected directly to the ion source of the mass spectrometer. And that ion source has a flow to it that it's pulling liquid down into it. And so the solvent there is kind of forming a continuous flow into the mass spectrometer. You can play with these flows to get different aspects out of the system. So if you overflow, we call that the overflow mode, and you kind of create this dome-like structure on the top of the probe. Or if you use less solvent, you can over-aspirate the system, and you create this basically miniaturized sink drain on the top of this probe, where the liquid, the solvent here, is actually moving very fast on the surface, and that helps actually take analyte as it comes in contact with the liquid and sends it right into this inner capillary that goes to the mass spec, and we can then get that really going pretty fast. The initial work that Gary Van Berkel and Vilmos Curtis did was back in 2017, where they demonstrated that we could use the I.DOT system to basically eject droplets into this OPSI probe that we built. And this proved to be very powerful. So by injecting small droplets, you could get reproducible peak features. So on the top right here are total abundance of a particular compound contained in the droplets ejected by the I.DOT system, where each peak here in the trace is actually a different droplet. So we're measuring several droplets sequentially. Basically, we just position the I.DOT system to eject that droplet directly into our open port. And the open port here is kind of on the bottom left, and it's made to fit right under the I.DOT system. And so you can change the rates in which you do this to basically enable some different experiments, basically akin to doing direct infusion mass spectrometry if you go fast enough. So for example, 1 second pause here ended up basically looking like a direct infusion experiment. And we also demonstrated that you can measure a variety of different compounds. You're doing, in this case, electrospray ionization. And so we can look at larger molecules like proteins. So this is an example of a mass spectrum of cytochrome C that was measured using this I.DOT open port system. And we can look at charge distributions that are common for these types of proteins and use that to look at the chemistry in detail from different analytes. That's not what I'm actually going to talk about today. I'm going to talk about our new and improved system that we developed starting in 2022. From the initial report to when we were actually able to get funding to continue to do more and more work with this, a lot of things changed, and we ended up having a lot of new challenges. With the newer I.DOT systems, we needed to develop an even smaller open port sampling interface probe, and so we kind of did that with this picture here. This is basically the open port on the left fitted onto a 3D printed mount that can sit on the lower panel of the I.DOT system. So it's kind of shown here on the right picture in B. And we also needed this to be able to basically operate on all of the different mass spectrometers that we have in our laboratories. So that included several SciEx mass specs and a thermo QE Orbitrap system. And so this I.DOT system, we were able to basically put on a movable cart, and we can use that cart to transport the open port to any of our mass spectrometers, so we can do any number of different mass spectrometry experiments that we want to with our capabilities. And then the last bit was that we needed to update the software to enable better control of the I.DOT system for our particular use case, which there's a number of of different particulars that we need to be very controlling of in order to enable online chemical characterization. And so one of the nice things is that we worked with the I.DOT folks to basically create our own droplet control software and kind of work out all of the different kinks that we ran into with the newer operating system. Most of them for us was that the I.DOT became kind of too smart for what we needed it to do. So the I.DOT operating system will try to use all 8 dispensing heads at the same time and make the most efficient route possible. And in our case, we needed to be very careful to eject droplets with a certain time between them so that our mass spectrometry signal doesn't overlap. And so using the API of the I.DOT system, we basically created this software to allow us to measure any well in however many replicates we wanted to do, and to also control the delay between dispensations of the I.DOT system. And so using this, we can basically create any sort of protocol that we want for measuring from a 96 well plate. And that was a big improvement for us to increase the speed of the actual 96 well plate assay, and then allow us to be creative in how we want to measure different samples. So the actual use case for us in this particular project, we're trying to engineer enzymes for nylon degradation. So as we're probably all aware that global plastic production keeps increasing, a lot of these plastics are very hard to degrade and costly to degrade. So developing new enzymes to enable their degradation is a path forward to maybe reducing some of this plastic pollution and recycle some of the plastic that we create. And so this particular project, like Leann talked about in her talk, has a we're iterating the design, build, and test and learn of a particular nylon enzyme that was found by the Nagoro group in Japan. And we're engineering this enzyme to be better for creating different nylon degradation products. In our particular use case, the Oak Ridge National Lab, we also have a manufacturing facility. And in that manufacturing facility, they'll use a nylon mixed with a carbon fiber to create a composite material that can be used to make all sorts of different 3D printed parts in various scales. And one of their pain points right now is that any of the waste that they generate, the carbon fibers are a very valuable product, and they would like to extract that out of the nylon polymer so that it can be recycled. And so that's kind of one of the use cases for us that we're targeting with this particular project, is to be able to extract back out these carbon fibers and recycle them and use them in this additive manufacturing process. So the particular analytical needs that for this particular project is we needed to have really high sensitivity because the current enzymes available that degrade nylon just don't work very well, right? So the current enzyme that we have converts about 0.0027% of the solid nylon that we put in. You need to have a decent sensitivity to be able to measure any of the degradation products coming out. The traditional chemical way of looking at polymer degradation is using gel permeation chromatography. And basically any sort of chromatography is going to be generally slow. So you're looking at maybe 30 minutes of runtime per sample, and sometimes the use of various solvents and things can be quite costly as well. And then, of course, we want to eventually measure site saturation libraries like Leann described, where you're measuring 1,000 enzymes in one round of engineering. And so you quickly realize that you can't use conventional assays like chromatography-based assays to look at something of that scale. We need to create something that's a high throughput chemical analysis platform. And then lastly, of course, we need kind of concrete data outputs in terms of being able to quantify exactly how much of the degradation products are being formed and what their chemical identity is. And mass spectrometry allows us to kind of answer all of these different key points. The I.DOT coupled with the open port mass spectrometry system allows us to do this. Basically, we're using the I.DOT system to dispense small droplets into the open port, where then those drops are mixed with solvent and analyzed directly by mass spectrometry. And so what we've been able to do with our newer system is to dispense as small as 10 nanoliters of liquid as an individual drop and get a readout within about 3 seconds per sample. For us, we always measure each plate in triplicate, which means that for a 96 well plate, we can measure that whole thing in about 15 minutes. In terms of the reproducibility and quantitative accuracy, on the right are 2 examples where we have a calibration curve spanning over several orders of magnitude for, I believe this is a propanol compound in water. And we can look at each of these individual peaks here is a single droplet event that is measured by the mass spectrometer. And so in terms of what we were able to see with these nylon degradation products, this mass spectrum here on the left is an example of the types of products that we can see from the enzyme reacting or degrading the polymer. And basically, we see a variety of linear oligomers associated with that particular nylon. And we can also see several cyclic oligomer structures that are often formed in the manufacturing process and so remain within the polymer and get released into solution over time. With the enzymatic hydrolysis, we actually see a lot of these cyclic oligomers get converted back down to a linear dimer oligomer. And so this was, for the NILC enzyme, this was one of the first results that the NILC will predominantly create this linear dimer oligomer that's indicative here of the enzyme working well. And so we can use mass spectrometry to basically measure all of these different chemical compounds at the same time with each of our assays. And then going back again, we really need to make sure that our reproducibility and consistency of the system is rock solid, because we're going to then be using this to measure thousands of samples as an assay. And so shown here is actually a measure of 12 96 well plates sequentially, where in every 3 wells, we have the linear oligomer from nylon 6 separated by no enzyme control, and then the linear oligomer from nylon 6,6, again separated by control. And so that continues on and on and on for all 12 well plates. And if you compare the first well plate to the very last one, well, plate number 12, they're virtually identical, and we have basically single-digit RSDs and variance across multiple plates. So in this particular data set, we're measuring a little over 1,100 samples, again, measured in triplicates, so you end up looking at about 3,300 samples. No crosstalk was seen between each of those individual droplets measured, and we didn't get any evidence of drifts, which is great. So it's working really well as a high throughput system. Again, we test the quantitative accuracy and our ability to withstand different enzyme buffers. So as we increase the buffer percentage dispensed by the I.DOT system, we see a little bit of matrix effects, which are matrix ion suppression effects, but we can reduce this if we go basically below 5 to 10%. And then we can quantitate really highly accurately across many orders of magnitude in terms of both all the different linear oligomer species that are produced by the degradation process. And so with this, we've optimized the kind of time, temperature, parameter space for this degradation process, and we can optimize those for different nylon polymers themselves. And we've been using that to kind of refine the assay further. And as we've gone on to test different engineered enzymes or different selected enzymes that should be active for nylon degradation, we can see kind of differences in the chemical structure. So on the top graph here is the signal that we measure from the dimer signal. So our NILC enzyme, which is the one that's been reported in literature, that predominantly only forms this dimer, whereas none of other enzymes do. But if you look at some of the larger linear oligomer products, you'll see that several other enzymes can form these larger products, but don't really form as much as the smaller products as NILC. So there is some chemical selectivity here that we can tune for in terms of what products we want to generate in the engineering process for these enzymes. We're just ramping up now to start doing many thousands of samples, but we've tested our diversity panel of enzymes for the generation of a variety of different linear oligomer products. And in our diversity panel of 96 enzymes, we found all sorts of different activity for generating these linear oligomer species. We're in the process now of selecting new enzymes to basically engineer further to really target the linear dimer oligomer for nylon 6,6. To kind of close out here, we also use this system for a variety of different projects at the lab. We have just started a soil chemistry experiment where we use derivatization reactions to monitor for things like ammonium concentration in soil samples. And we can use the I.DOT open port mass spec system to actually monitor these reactions over time. And so these graphs show the reaction of this diamino naphthalene conversion where it creates this NAT product when it reacts with the ammonium. And we can see that conversion happening over time, and we can test different concentration ratios of the derivatization compound using the open port system. And one of the nice things with this is that we're not modifying the reaction in any way. We're kind of taking that stock reaction and monitoring it directly using our system. So with that, I'll close out and just want to acknowledge our team. So on the mass spectrometry side, it's myself, Vilmos, and Dana Carper collected all this data. And then there's a whole team involved with the enzyme engineering project. that are shown here on the right. And then, of course, our funding was initially through the Laboratory Directed Research Program, which is internal funding at ORNL. But we also have primary funding from the Department of Energy to do a lot of this research. And then also a shout out to the DISPENDIX team who helped us with working with their API to create the software that really enables all this type of work. And with that, I'll hand it back over to you, Neela, and thank you for your time.
Thank you, Jack, for your insights. Our third and final panelist is Holger Armbruster. Holger is the Global Product Manager here at DISPENDIX. Holger leads the charge for our products, from identifying market needs to guiding our products through development and launch. For over 9 years, Holger honed his skills in life science automation at Beckman Coulter, creating customized solutions for complex workflows. He earned his PhD at the German Cancer Research Center, DKFZ, in cancer research. Now, Holger leverages this expertise to champion the voice of our customers and translate their needs into innovative DISPENDIX products.
Thank you so much for the nice introduction, Neela. And again, welcome everybody. So as Neela already said, I'm going to talk about the application of microfluidic technology for the miniaturization of drug discovery and protein engineering workflows. So I want to go a little bit into the details, into the technical details of the I.DOT, and then give some example use cases. As said, first I'm going to talk about the I.DOT core technology. So I'm going to give a little bit of explanation how the I.DOT works, and then I'm going to explain in which applications for drug discovery the I.DOT is used mainly. And then I do 3 use cases. So I'm going to talk about single-cell proteomics as the first case. Second, I'm going to talk about compound inhibitor profiling. And then last but not least, I'm also going to talk about a microarray, very similar to what Leann presented. The I.DOT core technology is in principle very simple, right? So we do apply pressure to a source well, to the top of the source well, and then we dispense liquid from a pore at the bottom of the well, which comes in different sizes ranging between 60 and 200 micrometer. So you can see the scheme here, as well as the pore and the dispensing of droplets in the animation. So the principle is very simple. Obviously, the magic lays in the detail. So it's really about the size and the structure of the pore, but then also about controlling the applied pressure very precisely, depending on the dispensed volume as well as on the liquid type. So what you've seen on the last slide was 1 channel. The nice thing about the I.DOT is that it has 8 channels, and each of them can be controlled individually. So that gives you a good speed, a very high speed. But also lots of flexibility because the I.DOT has a self-adjusting droplet size between 2.55 and 50 nanoliters, depending on the liquid and the total volume you want to dispense. So that makes it really super flexible. And also it's very fast with 100 droplets per second dispensing speed. The I.DOT comes in different configurations. So we've got the I.DOT S and I.DOT L, which work with 96 well source plates, and the high throughput device, that's the I.DOT HT, and that works with 384 well source plates. Both the I.DOT S and the HT have a source well capacity of 80 microliters, and the I.DOT L has a 500 microliter source well capacity. Regarding the target labware, the I.DOT is super flexible. We've seen a nice example from Oak Ridge with the custom target labware, but obviously the I.DOT also works very well with standard labware, including deep well plates. So the I.DOT also has one feature that I haven't mentioned yet. The cool thing about the I.DOT is that it monitors each and every droplet that is being dispensed from our source well. So below each of the source wells, we have a light barrier, and whenever a droplet passes the light barrier, it's being detected and registered in the software. So that is great for quality control, right? So you can create a report, but it also helps you to save runs when, for example, your source well is running out of liquid. Then the software would create a subsequent run where you could dispense the missing droplet. So again, that's a great feature process control, reporting, and also process safety. When we're talking about drug discovery, I think we are all aware that at this point, drug discovery is more than just screening, high throughput screening, which is shown here in the middle part of the slide, right? So obviously the I.DOT is like super well suited for screening application, and that can be high throughput screening, it can be high content screening, cell-based and biochemical assays, obviously, but then also assay development, optimization, as well as array-based screening. Beside this, like I would call like more standard drug discovery applications, we also work very well in the omics field, so NGS sample prep, real-time PCR, qPCR setup, as well as single-cell genomics and proteomics, and LC-MS sample preparation. And on the other side, we also address the synthetic biology field, right? So the I.DOT can also be used for gene synthesis, transfection optimization, oligo library management, site-directed mutagenesis, and gene transfection. And again, these are just some examples, but I think they are a little bit representative for the customer base we are addressing and the applications in drug discovery. As said, so the I.DOT is a super flexible system. I've mentioned this. It offers a high degree of accuracy, helps you to minimize your reaction volumes. You can miniaturize and by this save money and also increase your throughput. So it also offers a high degree of speed, and it supports really a high range of liquid classes. So we're going from viscosities around methanol of up to 65% glycerol stocks. It works really well for really sticky enzymes, but also goes down to the less viscous solvents that you may want to use in some of these workflows. So the first use case I want to talk about today is single-cell proteomics using LC-MS. What you see here is a simplified workflow to introduce you a little bit into this field. You would apply the I.DOT here in the first step, for example, for dispensing the lysis buffer into a target plate, which is then used for cell sorting or cell dispensing, right? So you would dispense your cells into this plate, into this lysis plate, lyse the cells. And then in the next step, you would add further reagents required for processing or indexing reagents. And with the I.DOT, you really achieve a high degree of miniaturization, which gives you a more in-depth resolution for the proteome research, and also is that it saves quite some reagents. This workflow you're seeing here is similar to what I've shown before. This is from a publication from Nature Communications in 2021, where the I.DOT was used to dispense TMT reagents, right? So the group here started with the single cell dispensing or sorting into 384 well plate containing already all the lysis buffers. Then the samples were boiled, degraded, and then the I.DOT was used to add the indexing or labeling reagents as said. And afterwards, samples were pooled, dried, and analyzed on the LC-MS. The group could really make good use of the capabilities of the I.DOT to miniaturize the reactions. Because so what they observed is that when downscaling the input material, the resolution, so that means like the detected proteins per cell do in fact increase, but that is highly dependent on the flow rate. So if you look at the first part of the diagram, we see that when dispensing 2,000 cells or 500 nanograms of proteins, the difference between the flow rates is not as high. But if you go to 4 cells or 1 nanogram of proteins, we see that the difference between like a flow rate of 250 nanoliters per minute and 100 nanoliters per minute is significant, right? And if we go to 100 nanoliters per minute flow rate, we see that we detect 313 proteins per cells. And when we compare this to like the higher protein content, that's a huge difference because here we only see 9 proteins per cell. So That means that with the I.DOT, these customers could really significantly increase the resolution. The next use case I want to present to you is the I.DOT used in compound inhibitor profiling. We've been working together with AbbVie Germany on an application node comparing the I.DOT both against manual and other liquid handling technology. What you do see here is the workflow I.DOT versus manual, right? We're starting with a library of 14 compounds, including references, which are prepared and then put onto the I.DOT. So first we do the pre-dilution in DMSO, and then in the end we do transfer the libraries, the dilution libraries onto assay plates, and we also add the assay reagents. In that case, that was an ADP close or cell viability assay. So if we do this with the I.DOT, it takes us about 18 minutes. If we compare this against manual, we're at 65 minutes. So that's a significant difference in timing, right? So the I.DOT really helps you to increase the throughput of this group. Here are the results of this experiment. So first, what we observed was that the I.DOT had a good finding rate against like the known historical data points. So the IC50 values they observed in the past, and it was very comparable to their standard workflow, so almost identical, and even better compared to other liquid handling systems. And also the variation, so the set value was really excellent. In some experiments, it was really superior against manual and other liquid handling systems. The data you see here shows that the variation, the set value was about the same. So all in all, the I.DOT could really reliably reproduce the historical data and compete well also and perform better than other liquid handling technologies. So summary here, so the I.DOT, as said, performed the low volume dispensing with a very good reproducibility. And on top of this, it was also much faster and had significantly less stat volume compared to other liquid handling technologies. What you see here on the left side is the comparison for dispensing the ADP Clo reagents, right? So these are the assay reagents. So we see that with I.DOT and the manual pipette, it takes about like 270 seconds, whereas the other liquid handler takes 600 seconds, so significantly longer. And interestingly, or not surprisingly, the I.DOT has like a very low consumption of reagents and that volume, right? So with the I.DOT, we do have that volume of like around 250 microliters, and with the other technologies, we see a that volume of 1.6 milliliters, so we can significantly save on reagents here. We have already heard about microarrays in the first talk of this webinar. Here's another example. So this is a publication from the Karlsruhe Institute of Technology, just very close by Stuttgart. That's the so-called droplet microarray. It also has a very similar working principle as the first example. So we're also working here with the hydrophilic hydrophobic surface to define or create very small droplets on a microarray. The droplet microarray from the KIT or AcroArray can be used for different applications. So one of the applications here that we see on the left side, this is testing of antibiotics on bacteria. So what we did here was to dispense bacteria using the I.DOT on the droplet microarray. And then we added antibiotics to it. And as you can imagine, we could really go very low regarding dispensing volume. And also we could hit the spots super precisely. So that was in this case a huge advantage of the I.DOT. And in the end, in this experiment, an optical readout was performed. So basically the researchers analyzed how the cells reacted to the different antibiotics in a miniaturized way. The droplet microarray can also be used for other experiments. So we've seen an optical readout. It can also be applied for MALDI-TOF MS and infrared spectroscopy. And in all cases, the I.DOT can be used to spot the required samples and reagents on the slide. So here in this case, for this use case, again, the I.DOT significantly reduces the reagent costs and speeds up the screening of a large amount of compounds on these arrays. So that's it from my side.
Let's go ahead and go to the question and answer portion. There are a couple great questions for Leann. The first question that I see is, do these droplets on a microarray evaporate?
Yes, they do. We did have some evaporation controls, some environmental controls to minimize that, but they can evaporate. They are stable for a little longer than you would suspect. You do have time to actually run with them and use them. Just like any liquid, they will evaporate.
Got it. And then how are you able to normalize the amount of DNAs for the fold improvement?
That's a great question. So the final fold improvement graph that I showed was with a higher production volume of DNAs that then was purified, characterized, and normalized, so that we could validate that the improvements we were seeing in high throughput were correlative to enzyme production, were correlative to specific activity, not just improved enzyme production.
Thank you so much. I'd like to give a special thank you to all of you for attending our webinar today, but especially our panelists, Leann Teadt from Codexis and Jack Cahill from Oak Ridge National Laboratory. No matter where you are in the world, we thoroughly enjoy connecting with the scientific community, and I genuinely wanted to make this a great experience for everyone. Have a great rest of your day, everyone, or night, wherever you are, and we'll see you in the next one. Thank you so much. Goodbye.